I try to give you my perspective about what is engineering, and I try to be a little bit provocative, trying to say that synthetic biology, or what we today define as synthetic biology, can actually lead for a new green revolution. So we heard Louis talk about the first green revolution, that without it we wouldn't be here today. We do not have the food that we actually can consume, but there is a dire need for another green revolution. So the problem is that human population keeps growing. It could be good, but it also could be bad. So these are the estimation by the UN about how human population could continue growing. And it could be that in the lifetime of most of us, we will reach the 10 billion score for human population. And the question is, how are we going to feed all of us? It was calculated that in order to be able to avoid global famine, we need to almost double agricultural production. So this is again an analysis that was made, which is quite nice. You see here how the yield, agricultural yield for the four main calorie sources for humanity were improved from the late 60s and up to 2010. You see that there is a consistent growth, but in order to double agricultural production, we need to follow this dashed line. And if you project from how we progress so far, this is this line. So we see that there is a huge difference between the level of progress we are currently sustaining in agricultural yield to that we actually need to do. And this is a big problem. Maybe not, we don't feel it here in Europe or in Northern America because we actually have too much food here. But in the developing world, this problem is already quite apparent. And even if we redistribute our food, which is, of course, another problem why people get hungry today because the food is not distributed equally. But even if we redistribute everything in an equal manner, soon there will be not enough food to feed everyone. So how do we solve it? A major factor that limits agricultural productivity is the low efficiency and rate by which plants use light to take CO2 from the air and fix it into biomass, in this case, sugar, that from which biomass can be derived. So the details here are not important. What's important is that what plants are doing, basically, they use light to split water and take CO2 from the atmosphere to convert it into biomass. And this process is quite an inefficient process. Now here you might wonder to yourself, plants have evolved for millions, hundreds of millions of years. Plant breeding, as we are doing in our culture, is being done for thousands of years. So here I give, for example, the famous Mendel experiment, but of course we are doing plant breeding since we discovered agriculture, which means thousands of years. So one would expect that during this very, very long time cycle, carbon fixation will reach optimality, that there will be nothing else that we could do in order to improve it. So how come the process is still so inefficient? And what I would like to claim, that what plant evolution and plant breeding has in common is the fact that both of them work as a tinkerer rather than engineer. And this is based on the famous quote that evolution itself can be viewed as a tinkerer. And many of you might ask, what exactly is a tinkerer versus engineer? So let me explain, because I think this distinction is essential for our understanding of what we can and cannot do using rational design of natural processes. So a tinkerer is a guy or a girl or whatever that takes an existing machine, existing system, and play with it, change it. You can make it a little bit better, a little bit different, but the starting point is a starting point of an already existing system. And this is, of course, how evolution works. So you have an already existing system, let's say a bacteria, and this bacteria evolves to do something a little bit different or something a little bit better, and throughout the ages, it changes and changes and changes until it reaches the situation that you have a human being standing here and human beings sitting there. But this is not an engineer, because an engineer starts from scratch. Engineer takes the design principle of the system to understand what would be the optimal solution for a given problem. So basically, if we look at it from the point of view of how to reach optimality, how to reach maximality, the point of view of a tinkerer is very limited. A tinkerer can see only its immediate surrounding, so it can climb a mountain because it sees the immediate surrounding, which part is becoming higher and higher and higher, but then it gets stuck at a local maximum. While an engineer looks at it from a bird's view and can actually see what is the local optimum that can actually solve the particular problem in the best possible manner. Therefore, an engineer has always an inherent advantage over a tinkerer, and this is why, despite of millions of years of evolution and thousands of years of plant breeding, we still have a lot to improve when it comes to carbon fixation. But now we have a problem. Even if we accept that engineering is the right approach, we cannot just engineer biological system as we engineer a mechanical system. An electrical engineer, a mechanical engineer, know exactly all the parameters that governs his or her system. And therefore, he or she can design a really good solution. However, biological systems are chaotic. By chaotic here, I mean the scientific definition of chaotic. So everyone here probably knows the cliché of a butterfly clapping its wings in one place, and in the other side of the world you have a hurricane. But of course, this cliché also holds a grain of truth. Basically, a chaotic system is a system which is extremely sensitive to changes in its input. And according to these minute changes, huge changes in the output or in the behavior arises. And of course, engineering such a system is very difficult. How can you exactly predict what is going to happen to your system if you know that if you change it a little bit, every small noise can be amplified? The trajectory, this chaotic trajectory, also prevents us in some cases to understand the causality within the system. Why did we get a certain behavior when it did this or that? Okay, so how do we do it? How do we engineer a chaotic system? First, you need to not be very vain and say, don't say to yourself that you know the optimal answer and there is only one answer that you need to try to implement, but try to think about multiple solutions or engineer multiple solutions, knowing that even if one fails, you still have the others that you can rely on. Second, you want to try and focus on parts of the system which are less chaotic than the other. I will give you an example in a few seconds, but just think about it. In an entire system, there are always parts which are very connected to all other parts, and then they tend to be very chaotic because every change you make to them will influence the entire system, while other parts of the system could be poorly connected, and then they will be more stable. And finally, and probably the most interestingly, is that if you really want to be a good engineer, you should also use the gifts that nature gives you and the tools that nature is using itself. And this, of course, requires a lot of explanation, which I will give you in a second. But I would want to shift now to actually go back to the question that I raised of how to engineer better carbon fixation and how we implement these logic into carbon fixation. So as said, the natural process of carbon fixation is quite inefficient, and we need to engineer a better solution. If we look at the carbon fixation process, I give you here a schematic way to look at carbon fixation. Each arrow here represents an enzyme. Enzyme is a protein, it's the machinery of the cell. Basically, we are all composed of enzyme doing metabolic work that enable us to live. The stars represents the compounds in the cell, so each enzyme converts one compound to the other, and another compound to the other, and so on and so forth. As you can see in this process, CO2 is being assimilated, and there is some biomass precursor that is being produced that enable the plant to grow and multiply and so on. Now, if we try to change the carbon fixation pathway itself, this would not be a smart idea because this part of the system is it's highly connected to all parts of plant metabolism. It's a very chaotic system. So this is exactly not the part on which we would like to focus. So instead, we decided to focus on another process which relates to carbon fixation, which is called photorespiration. So photorespiration is an essential process. You must convert this product back into the system. But the problem of natural photorespiration is that it releases CO2. And this, of course, directly counteracts what we would like to do, because we would like to fix CO2, and here we have a system that actually releases CO2. So we came and thought how we can solve this problem using engineering tools. So we designed pathways, metabolic pathway that contains new enzyme that would be able to bypass photorespiration without the release of CO2. And this means that you keep much more carbon in the system, and this could support the growth of the plants and boost agricultural productivity by up to 60%. So this is really, a true engineering approach. And it's also a true engineering approach because we thought about this enzyme not by thinking about existing solution, but actually really designing stuff or processes from scratch, a de novo engineering. So how do we do it? How do we engineer the missing enzyme that will be able to support this process? So here come the enzyme engineering logic into place. So what enzyme engineers are usually doing, they are taking existing enzyme and they change them rationally so they will be able to make a new process. So basically, this is funny. What the enzyme engineers are doing is similar to tinkering because they start with an existing enzyme and tinker it to make a new enzyme. So it's a combination of the engineering and tinkering. Okay, the problem is that enzyme engineering can take us not very long or not very far because it's a very laborious process. As you can see here, there are many steps. It's very costly. It's very tedious. Every cycle of this it takes you a lot of time and money. Also, testing this pathway directly in plants is also very tedious because plant metabolic engineering is still a challenging process and it restricts the amount of how many cycles you can check. So what we need is a technology to enable us to take this unevolved enzyme or still rather poor enzyme and be able to optimize them in the most efficient manner. And what we are doing, what we specialize in, is using bacteria as a platform to take an unevolved pathway, to improve it and optimize it before we then can express it in plants to support a better carbon fixation rate. So you might ask yourself what bacteria has to do with all of the process because bacteria is not doing photosynthesis. So let me explain to you by focusing on the bacteria. Bacteria usually converts its food, usually sugar, into essential cellular components that enable them to grow and multiply. We engineer the bacteria such that it cannot produce one of its essential components anymore. And then we introduce the new pathway as the only way to produce this essential component. Therefore, we couple between pathway activity and bacterial growth. No pathway activity, no growth. Low pathway activity, low growth. High pathway activity, high growth. We grow the bacteria for many generations in just a few weeks, something we could never do with plants. And during this short life cycle, fast evolution takes place. Changes to pathway enzyme emerges spontaneously, and the best pathway variant arises naturally as it outgrows its competitors and take over the bacterial population. So basically, while the project is starting as an engineering project, something we designed de novo, we let nature do the improvement by using nature own tricks, which are, again, natural selection. Okay, so the last thing I want to show you is a little bit how we do all of this bacterial growth. And this you can see here, I hope at least, yeah. So this is probably the smallest place in which we can do a growth experiment. Each of... Okay. Sorry.
Sorry.
Well, you know,
I hope it's working now.
Do you hear me? Yeah. Assuming. Okay. So So let me show you a little bit. So this is the smallest place in which we can actually grow the bacteria. This is called 96 well plate. So each of these holes is a well. In each of these well, we can put 150 microliter, which is a very small amount, of cells. And you can see that we can grow 96 different bacteria here at the same time in a machine that checks it all the time and control the temperature, so we can characterize all of these strains simultaneously. On the other hand, the biggest machine that we are using is this one, and this I think is especially interesting. So here is when we are doing the evolution. I said that we put the strain that we want to evolve for higher pathway activity and grow them for many generations. This is the device. So basically, we put a culture, a medium, and this part, containing the cell. Here there is a small propeller, I hope that you see it here, that makes them all the time moving, so they'll get aerated, air will go inside. And you can see here all the complex, it looks a bit like a spaceship, all the complex input and outputs that goes into the bioreactor. So this is the fresh media that we are putting in and the old media going out, and this could sustain continuous growth of the bacteria here for days, weeks, months. During this time, we can actually do the evolution and get for higher pathway activity. Thank you. And Helena, will you join me?
Interesting talk. So we're both scientists, and I always find it very interesting to know why people are doing what they're doing. And so you're working on carbon fixation and feeding the world, which is one of the biggest challenges that we have as a species. And so why did you get interested into this topic? Is it because you were interested in biological organisms and then you saw this process of carbon fixation that you found was interesting? Or were you driven more by a solution, like a social problem that you were trying to solve, and then you found a way to maybe help into that direction?
Yeah. So I was always applicative in mind. So I always was oriented to try to solve problem. So there is, this inherent tension in science between those that focus on basic research and said that the applicative part is, secondary and it's not the real interesting part, and those that see the applicative part as, as leading the way, and the basic research actually follows that. So I was always in the second camp, and I don't think that there is right or wrong here, of course, but for me, it was always trying to solve problem. And I remember, in my case, from high school, that my biology teacher accused me that I'm not a real thinker like a scientist, but more like an engineer. And it's true, I'm thinking like an engineer, but it doesn't mean that I'm not doing science. So and what about you in this regard? So do you see yourself more as a scientist or engineer?
Well, I went to engineering school, so yeah, I was formatted to think about solving problems, which I found was interesting, and I always being interested in health problems. I remember that when I was a kid, both of my parents got really sick. They got like a nasty infection, like a bacterial infection, and like from one day to the other, both of my parents were in the hospital, and that was like a real shock to me, and so. I got interested in diseases and this kind of stuff and trying to solve these diseases. So I saw that as a problem.
So it's a little bit not fair, isn't it? Because it's two engineers sits there, no one here to represent the basic science.
Yes, but I mean, the work we're doing wouldn't be possible without the basic science. And I think it's a little bit lying to ourselves to say that they're not like engineers and we're not the fundamental scientists, because whatever we're doing, we're learning about how the plant works or the bacteria work. So it's like a constant feedback between those.
I think that you see it probably as well, but so we scientists, we live by funding. Basically, we cannot do anything by not getting money from the public. That's, money from you, basically. And there are some funding agencies that want to see almost only applicative stuff. The European Commission, for example, if the European Commission is funding something, it's always needs to have some, a very clear output how we're going to change humanity, while other funding agencies are focusing only on basic science. So, all of us are doing both of them.
Yeah.
Even from the point of view of getting money, you need to be very smart if you put yourself as an engineer or put yourself as a basic research kind of scientist.
Yeah. So now, in terms of feeding the world, we're talking about seeds, we're talking about plants, about evolving plants in agriculture. Do you think actually agriculture is going to solve the problem of feeding people?
Yeah.
Or?
So, that's, in a sense, this is the $1 million question. So we definitely need to have better agriculture, that's for sure, or at least that's what I'm trying to propose. But I think that if we want to be a little bit crazy and think about, the future, we could think about other ways in which we could feed ever-growing population, not just here on Earth, but maybe also when we are going to adventure out of Earth. And this could be actually, and it will sound crazy to some of you, eating microbes. So some microbes are actually very nutritious. Yeast, for example, people eat yeast today, and it has all the necessary building block in food. And another thing that we are trying to do in my lab is actually to enable microbes to grow on electricity and CO2 as the only energy and carbon source. So basically, we can convert wind and CO2 into food, and then we can feed the world in other ways but agriculture. So you can imagine that if we are going to Mars in 50 years, instead of relying on the contaminated soil there, we can actually try to produce our food from microbes.
Scratch, from microbes.
Yeah. So hopefully we will not be married to plants forever.
So what you're saying is basically that if we want to survive as a species, we will have to really change our whole alimentary culture or of food production.
I think so. I think that's true also today. If you look,
we have no choice. We need to eat GMOs or people are scared, right?
This is the tricky point, of course, the GMO. This is, talking about GMO is also being a little bit provocative, and, people probably in the audience has completely different views about it. From my perspective, as someone who's actually in the field, I think that some of the fear related to GMO are founded, but many others are, I would even say, related to a politics of fear. That, if you see how the world is going on in the last decade or so, you just try to make people feel of this or that. So many of the issues that make people, take a step back because of GMO related to a bit of ignorance about the field. Because from the perspective of what we actually thought that could be problematic, we currently have no scientific evidence for this problem actually occurring.
Yeah, so it is true that the general public is unnecessarily educated on the inner workings of the GMO and whether it affects them or not, but I think that their fear, it's founded in the sense not of the molecular level, but on how this technology of the GMO is being used and all the political and social problems that it has in hand. So indeed, the technology can have a lot of benefits, and I agree with that, but it's the way it's been used right now by certain corporations that is really provoking this sort of not necessarily fear, but rejection of this technology. And so in that sense, I think we also need to be a little bit critical in, whenever we develop a technology, how are we actually going to implement it and whether it's going to damage or not different levels of society. And not only on the molecular level, of whether when I eat a GMO corn, I'm gonna get sick, which there's still for now, no scientific evidence for that. But, people care that it might affect the farmer or it might affect
So here I fully agree. So here I fully agree. I think that the major problem we have currently with the GMO that it is controlled by very big companies that abuse their power in many cases. But this is a general problem that we have currently in the Western society of, big companies that controls big sectors. I think that it's important, though, to distinguish between the technology itself and those that control the technology. And I would agree that we have a lot to go to make this technology something that is available to everyone and not controlled by big company that creates social disturbances. But this is not to say the technology itself is bad, just the way that it's come. Exactly.
It's the way we use it, yeah.
Yeah.
And well, I now want to move to this analogy that you were using about biological systems as chaotic systems. It's funny because you say that they're very sensitive to changes, like noise gets expanded. I actually believe they're quite robust. I actually believe that it's different from my experience with biological organisms. Whenever we try to change them, they will come back to their initial form, or they will basically escape our control. And in the case of your plant, so imagine that you put your plant outside in nature. I think there is a reason why their plants are not that efficient in carbon fixation. Do you think that at some point, well, the plant will lose that ability? Like how? Like
So
I think they're quite robust.
Okay, so you're right that living organism can deal with a lot of different conditions and survive, and actually not just survive, actually grow and multiply in many different conditions. I would claim that for changes that the organism was evolved to cope with, it will be very robust. So a plant could survive a long drought conditions, as we will hear later. A plant could survive different level of fertilization, of different level of light, and so on and so forth, because the plant was evolved to actually survive it. But if you are go inside and tinker with its small genes, with how it works from the inside, this kind of abuses the plant was never actually evolved to withstand. And this is where the chaotic system comes into play.
Yeah. Well, in my case, I engineer a system, and then since basically I'm provoking something to happen, the bacteria that I engineer lose their ability after a few generations.
But I would call it chaotic response also. Because basically it's exactly the same point. You engineer a system, you think that you know what's going to happen according to your knowledge of the system, but eventually the system behaves how it wants to behave, getting rid of the genes that you put inside, or changing its metabolism, its overall genetics in a way that you cannot predict. So basically this is exactly this kind of a chaotic behavior that, you cannot just, design stuff and it will work like an electrical engineer designing a chip, and this chip is just functioning by itself.
Yeah. So going back to your plants, so if they are able to do carbon fixation more efficiently, what does it actually mean? Like, is the plant going to be bigger, or is it going to be like? I have a hard time.
That's a great question. The real answer is don't know.
Okay.
So it really depends on the plants.
Yeah.
So there were very interesting experiment done with plants that you put under elevated CO2. For example, you put them under CO2 level which are triple the amount of we currently have, and this of course makes carbon fixation much faster. Okay. Some of the plants grew very fast, but then stopped at the same amount of biomass. Some of them grew in the same rate, but the amount of harvested biomass, the amount of fruit of the grain was much higher, and some of them did not change at all. So basically, it's really dependent on the plants. And it's again, saying about chaotic system, every plant, every system, every context behave a little bit differently. So we don't know yet what increased carbon fixation rate will actually translate in the field. It could be that for rice, for example, it will increase the grain. It could be that for wheat, it will not do anything. So this is simply something that we need to test and see.
We will have to live with the contingency of
yeah.
Okay, thank you.
Thank you.